Well, I want to first reiterate something that Merlin said before. I've been attending this meeting since its inception in 2000. So as an undergrad at Purdue, La Adiola brought me to this meeting. So again, speaking to all the graduate students, and if there are undergrads out there, this is a place where you can grow up, where you can make those connections. All right? So what I'm going to talk about today is just a new line of research that's bringing together these prenatal effects and ultimately postnatal nutrition. And connecting with what Bill said, I'm going to tailor this talk to the production animal ag side. But the other half of my lab uses the baby pig as a biomedical model for looking at human health. So this small pig that we're going to talk about today, we've also looked at it from a cognitive development standpoint, and it is a great model for human infants. So implications here not only on the production side, growing pigs, but also on the human side, looking at brain development. We also look at iron deficiency in the baby pig. Again, ways of connecting and providing basic science which can then support what we're trying to foster with animal agriculture. So intrauterine growth restriction, and specifically providing support to these baby pigs by the provision of arginine, is what we're going to talk about today. Now this is an age-old question. If we can somehow reduce pre-weaning mortality, this is going to have direct economic benefits for the swine industry. So we know there are a number of different factors associated with pigs and why they may not survive through weaning. And I've listed 10 of them here that came from a review article by Lay and co-workers in 2002. And a number of these factors are being affected by the uterine environment and are going to be interrelated. For example, if we have a large litter size, we have potential for dystocia. This may lead to low birth weight piglets. So what we're going to focus on today here are specifically piglets, the phenotype that we're actually seeing in production, a low birth weight piglet, and how we can use nutrition to supplement that piglet during the pre-weaning stage to ultimately affect growth and looking at metabolomics as well. So I'm going to start off by just a couple of definitions. There's a lot of acronyms that are used here, but ultimately, if that fetus doesn't receive enough nutrients when in the uterus, it is this intrauterine growth restriction, or IUGR, that's ultimately being manifest as a small pig. So here I'm using the term low birth weight piglet, or LBW, for that small pig. So relative to its litter mates, it's got a small stature, but overall we're talking about healthy pigs. They have no other complications when it comes to clinical outcomes. These are healthy but small pigs that we're talking about. The other term that's used in the human literature is called SGA. So a human infant who's born small for their gestational age. This is the equivalent on the human side. And again, this is where our piglet model is going to cross over. So we can study cognitive development using these small pigs, and they do have the same deficits in white matter tracts that we see in human infants. So providing the basic science, just translating between these two. So here we're going to talk about low birth weight pigs. Now there's a large body of evidence out there to suggest that if insufficient nutrients get to that fetus, that between 15 and 20% of those piglets could be considered as low birth weight. And Wu and others define this as a pig weighing less than 1.1 kilograms. Now more recent evidence suggests that up to 25% of all pre-weaning mortalities may be associated with piglets being born small. And if we look at, and that's for a variety of reasons. I mean, thermal regulation, they're going to get cold. They may not be receiving enough milk early on. So there's a number of reasons why they may succumb to mortality in the pre-weaning phase. But if we look at the USDA National Animal Health Monitoring Service and some stats, the most recent stats there, 46% of all pigs being laid on by the sow, an additional 23% dying of starvation. These are producer survey data that are coming back. But here, I think you can agree that nutrition of both the sow and the piglet are going to be involved in many of these different reasons why we would have pre-weaning mortality. Now, as far as characterizing what that low birth weight piglet is, this has also been done from a functional perspective. And we know that there's abnormal development in a number of different organ systems within that small pig. And this is especially true of both the liver and the small intestine, which are important for us as nutritionists. So in general, these low birth weight piglets are going to have reduced absorptive area in the intestine, which means they're not able to extract nutrients from the diet as efficiently. Additionally, they tend to have a leaky gut for a longer period of time. So we have compromised intestinal structure, meaning that we're getting not only nutrients, but also bacteria that are going to pass through. So increased incidence of disease because of holes in the gut, if you will. Additionally, these pigs inherently have a compromised immune system. And this may be related to hypoxia, depending on their birth order, if they were the last pig being born, but a number of different factors that were either happening in the uterus or immediately after those pigs were born that's preventing them from mounting an immune response effectively. Now, bringing this back to swine production, what we see in the barn from these small pigs is on average about an 8% lower growth rate all the way to market. And in general, what that means is an additional 12 to 23 days to get these pigs to market weight, which of course is going to reduce profits because we need much more feed to get them to that point. From a biological standpoint, if we look at the muscle structure of these low birth weight piglets, they're also different fundamentally in the way that those muscles have developed prenatally. So they generally have a lower total number of muscle fibers. And if we look at the structure of an individual fiber, it has a larger cross-sectional area. Being married to a meat scientist, I'm told what this translates into is poor meat quality. So all of a sudden we have pigs that are born small, that if we can get them to market, we're going to put the money in to get them to market, they still have a lower meat quality. So this is an issue from our perspective. Additionally, the literature is a bit conflicted on fat deposition in these animals. Some show that a piglet born with a small birth weight is going to have a larger thickness of back fat, but this is not quite clear in the literature. It's just something that's been seen. Now, the way that nutrition ties into this is if we can provide supplemental nutrition, if we can somehow assist this pig during the pre-weaning period, then perhaps we can reduce mortality or increase growth during that time. So it's trying to develop a strategy from a nutrition and management perspective. Where we chose to focus was the role of arginine in neonatal nutrition. Now, arginine as an amino acid is considered a conditionally essential amino acid, but in this case, with the pig growing so rapidly, there are fairly high requirements for arginine. And what we've studied here is building upon the literature that's coming out of Texas A&M, a paper started by Kim et al. In 2004, where they took baby pigs that were raised artificially. They took the sow milk replacer, which should have been adequate, and they added additional arginine on top of that. What they observed was that they got increased growth in those pigs, which suggested that arginine was a limiting nutrient for that baby pig. Now, if we look at the arginine content of sow's milk as a percentage of crude protein, there's quite a range in the literature. A twofold difference from the highest value of about 5.5% all the way down to the Kim et al. 2004 reference, which is about 2.8% arginine relative to crude protein. So quite a large range, on average around 4.4 grams of arginine in that milk. Now, providing arginine in the milk is only one way that the pig is receiving this arginine, because we have endogenous synthesis as well. So when we go through the calculations of arginine flux in the piglet, on average, a pig is ingesting approximately 1 gram per day from the diet. Additionally, that piglet is able to produce an additional 1.7 grams per day through the coordinated action between the kidney and the small intestine, since the liver will continue using that arginine for urea. So there's a net synthesis of arginine coming out of the intestine then. So this means a total arginine requirement of about 2.7 grams per day. And what these authors noted was that dietary intake is only 40% of the total requirement. So if we can supplement, if providing arginine allows greater growth, and we can supplement in that way, then perhaps this is more of a limiting nutrient than first suspected. Now, we're all aware of the arginine functions in the body, but first and foremost, in that pig that's growing so rapidly, incorporation into muscle protein is going to be very important. So for lean tissue accretion. But certainly as part of the urea cycle and handling nitrogen in the body, arginine is very important, as well as production of the second messenger in nitric oxide. So both being used as a vasodilator and also important in the immune response. So a number of different factors where arginine is important, all of which the baby pig that's born small could be exposed to. So with all this in mind, we wanted to test the hypothesis that providing oral supplementation with arginine to these low birth weight piglets could actually improve growth performance and rectify any metabolic problems that may be occurring with the low birth weight piglet. I want to highlight the word suckling here because this is how our study is different than previous research, that we were actually leaving these pigs on the sow during that pre-weaning period and simply providing them with an oral supplement twice a day. So we broke this down into two objectives. First, we wanted to know what is the low birth weight piglet on our farm. So that first objective, defining these criteria for both birth weight at our UIUC swine herd. The second then was the dosing study, where we're actually identifying pigs that are either average birth weight or low and dosing them with arginine on a daily basis to look at growth performance and metabolic outcomes. Now, it's almost sad to say that the entire first objective can be summarized on this one slide, but this was started as an undergraduate project. What we did was we went out to the farm, and over a 3-year period, we entered farrowing records. So these are birth weights of individual pigs collected between July 2008 and October 2011. This data set represents a little over 8,000 piglets. So it was an undergrad's job to type in all these numbers, verify all these numbers. What that allowed us to do is to produce this histogram. This is a histogram. On the y-axis, we're looking at the frequency of birth weights. On the x-axis, we've taken individual pig birth weights and binned them at 1/10 of a kilo for each of these bars. This exercise then allowed us to identify what is an average birth weight piglet here, denoted in the 3 dark blue bars in the middle, and our low birth weight piglets in the lighter blue bars over here. So our low birth weight piglets, by design, we looked at the lower 10th percentile of our population. So this represented, in this case, 9.3% of all pigs, and we defined the low birth weight pig as a healthy pig weighing less than or equal to 900 grams. So a little bit different than in the previous research where they looked at it at 1.1 kilos for that LBW pig. Our average birth weight then was again a healthy pig averaging between 1.3 and 1.5 kilos. And in total, this represented 40% of our total population. So now we've defined what is our normal birth weight and what is our low birth weight piglet. With that in mind, we went on to the dosing study. So having those criteria in mind, we selected healthy littermate pairs. A total of 16 litters were used, and these pigs were selected after processing, so at 24 hours of age. We only selected litters that had both an average birth weight and a low birth weight piglet within, and they needed to be both of the same sex. So we have equal numbers of males and females enrolled in this study. So after processing, when those pigs were actually put onto the study, they remained with the sow throughout weaning. And what we did is we weighed them on a daily basis in order to calculate the volume of amino acid supplement that they would receive. So we used the same dosages as the earlier work by Wu et al., that we were providing L-arginine HCl at 145 milligrams per kilogram body weight per day. And we were using L-alanine as a non-essential amino acid and providing this to be a isonitrogenous dose of that amino acid. So all pigs were dosed every 12 hours throughout this 16-day study. And apart from when we were weighing them and dosing them, they always remained on the sow with their original litters. So this is a color scheme that will remain. We've got average birth weight piglets that receive either alanine or arginine, orange and light blue. Low birth weight piglets receiving either alanine in green or arginine in dark blue. So this was a 2x2 factorial arrangement of these two treatments. We had a total of 8 pigs per treatment. And at day 16, in addition to collecting body weight gain over the entire time, we're also collecting blood to look at overall metabolic profiles. And I'll explain what metabolomics is in a second. So we'll jump right into the results. If we look at growth performance over that time, on the y-axis, we're looking at body weight of individual pigs. On the x-axis here, days of age during the 16-day study. So at the beginning, these are our pigs averaging 1.4 kilos for our average birth weight piglet and 0.9 kilos for our low birth weight piglets. What I'm showing here first are the pigs that were dosed on a daily basis with our alanine control supplement. We can see that at no time did these low birth weight piglets ever catch up or even narrow the gap between their larger litter mates in this case. So if we look at average daily gain, about 222 grams for what we consider our true control pigs, about a 32% reduction in our low birth weight pigs that received the same supplement. If we then layer on top of this the other two treatments, which include the arginine supplementation, here we've got our average birth weight pigs alanine, average birth weight with arginine. We've got about a 16% reduction here due to daily supplementation with arginine. Again, isonitrogenous daily intake of alanine and arginine in this case, so this is an effect specific to arginine here. In addition, in our low birth weight piglets, we get about a 16% reduction, and overall, low birth weight piglets receiving arginine, a 50% reduction relative to our average birth weight alanine dosed pigs. So in neither case did arginine provide any support to that pig or ameliorate any of that growth response that we were seeing. In fact, the same occurred in normal weight pigs. Now the other aim here was to look at the overall metabolic profile, and we used a technique known as metabolomics, which is an up-and-coming technique. Instead of looking at a specific panel of metabolites, this technology allows us to look at all metabolites simultaneously. So this uses bioinformatics. We're getting very large data sets. But the metabolome here is defined as that complete set of small molecule metabolites present in any biological sample. So here, our biological sample is simply blood at day 16. What we're doing is we're analyzing all of these metabolites simultaneously, so we're getting a broad overview of all of metabolism in one shot. Now there are companies out there like Metabolon, where we were able to submit these samples to that can provide the metabolomics service for you. So what they're doing is they're taking the blood, they're extracting it, and running it across a number of platforms, both liquid chromatography and gas chromatography, to separate all these molecules and identify changes due to our treatments. Now in the remaining slides, what we're going to talk about is how did our treatments affect these metabolomics, and these are all in relative fold changes. So our calibrator group was that normal weight pig receiving alanine, and everything will be expressed relative to that group. So in total, we were able to identify 323 metabolites. These are named metabolites. We have used standards to identify that they are correct. And in general, here's the number of metabolites that were affected by the main effect of birth weight group, the main effect of amino acid supplement, and the interaction of those two groups. So with a probability of 0.05, 46 metabolites were different between the average birth weight and the low birth weight pigs. Some of these went up and some of these went down, but a total of 46, there was a significant main effect here. The same for the amino acid supplement. What we're going to focus on today were those metabolites where we observed a significant interaction between low birth weight and arginine supplementation. And because these are very, very large data sets, I wanted to break this down into general categories where these metabolites fit into. So the interactive effects here were mainly involving the amino acids, energy metabolism, lipids, and the nucleotides. And that's what I'm going to focus on here. So the remaining graphs are going to be set up in a similar way. We've got two different metabolites on each graph, the same color scheme as before. Our average birth weight pigs receiving daily alanine doses are shown in orange, and these are set at 1.0. All other bars then are relative fold changes compared with that orange bar. We've also got the statistics shown here, and we're going to start off by talking about this is arginine in the blood of these pigs at day 16 after they've been dosed for that entire period, and alanine as well. Basically, the story with arginine comes down to when we dose pigs with arginine, we see more arginine in the blood. So this is persisting even though these pigs had been food deprived prior to collection of blood. We're seeing about a 1.5-fold increase in the average birth weight piglets that were dosed with arginine and slightly less in the low birth weight piglets. But no differences here between the low birth weight piglets receiving alanine and the average birth weight piglets here. In terms of alanine, again, the other amino acid that we dosed, no significant effects overall. And this is likely due to its role, its inter-organ transfer. We're not seeing many differences in alanine. So this was completely unaltered by supplementation or birth weight group in this case. Other metabolites that fit into this amino acid category that were most significant here included glutamate and urea. So glutamate, we saw a little over 1.5-fold increase when we dosed with arginine, and we were interpreting that as basically a glutamate sparing effect in these pigs, knowing that arginine can be converted into glutamate, therefore. We're getting an increase there because we also saw an increase in the amount of arginine in the blood. Now what's interesting is the low birth weight piglet dosed with alanine also had the same level of glutamate. So about a 1.6, 1.7-fold increase in blood glutamate concentration, even though they were not dosed with arginine in this case. However, our significant interaction here is coming from the fact that arginine dosing actually reduced the blood glutamate concentration here. So it tended to bring that glutamate concentration more in line with the average birth weight control pigs. The story continues with blood urea. So also involved in nitrogen handling in the body. Absolutely no effects here due to arginine in the average birth weight piglet, but a significant effect of birth weight group, as we had approximately 2-fold increase in blood urea in any pigs that were born small. So these low birth weight piglets on alanine, about a 1.5-fold increase, and an even larger increase when they were supplemented with arginine. So in general, we observed problems with nitrogen handling when it comes to these low birth weight piglets. May not be a big surprise, but what we're trying to piece this together then with all the other components here, metabolites, and make a story. Now, knowing that those low birth weight piglets had a lower growth potential, one thing that popped out of the data set were circulating nucleotide concentrations. So they have lower growth potential, lower lean accretion, and DNA synthesis could be suggested. And in this case, we're seeing highly significant interactions here for both cytidine and uracil. So no main effects, but interactive effects here, in that an increase due to arginine supplementation in the average birth weight pigs, a further increase when those pigs are born small. And in this case, by dosing with arginine on a daily basis, we're actually correcting that nucleotide concentration of both cytidine and uracil back to the same level as our average birth weight pigs receiving alanine. So for many of the metabolites, this was true. Arginine was able to correct the metabolic abnormalities. In terms of energy metabolism, glucose, we saw significant main effects of amino acid supplement and also an interaction here. We have an increase in these low birth weight piglets, and again, dosing with arginine has reduced that level of glucose down to our average birth weight controls. And the same was true not only for glucose, but also many of the TCA cycle intermediates, with malate being highest in this case in the low birth weight piglet, a 2.5-fold increase in that case relative to those same pigs that were dosed with arginine. So just to make a couple conclusions here, probably what's most important from a production standpoint is obviously growth. And in this case, arginine supplementation did not at all correct the growth trajectory of these pigs that were raised on the sow. In fact, by doing these daily oral dosings of arginine, it's actually negatively affected growth in both sets of piglets, which is troubling. So in our mind, this suggests that arginine was not limiting for these baby pigs, that providing that daily supplement was not helpful in this case. And so we need to go back and reevaluate how our study fits into the context of what was done previously, where they did see positive effects. From a metabolite and biochemical perspective, it was interesting that arginine supplementation was beneficial in terms of aligning those profiles. However, we can't sell pigs on a metabolic norm. So even though we're seeing a correction of these metabolic abnormalities, it's still not affecting growth that we observed in these pigs. In general, these pigs have appear to have issues with nitrogen handling, so especially with that urea. And that's something, an area that we'd like to look into further. And overall, the data suggests that the pig is shifting from using fatty acids as a source of energy to carbohydrates during this time. So the implications of this research, we believe it is possible to develop management strategies. However, the use of arginine does not appear to be a directly applicable one. So it certainly can mitigate those metabolic abnormalities of the low birth weight piglet, but we feel that focus should still be given on trying to maximize gain and minimizing profit losses due to the extra time that these pigs require to get to market. So looking into strategies like compensatory gain, for example, somehow reducing the time to market in these pigs. And with that, I would like to acknowledge our farm staff for providing access to lots of data records. Also members of my lab, including those who were assisting on the writing this paper, Dr. Nicola Sereo, who was instrumental with the statistics, and our College of ACES James Scholar Program. As I mentioned, this started off as an undergraduate research project, and it's gotten very, very large over time, but we do appreciate the support from our college. And with that, I'd be happy to answer any questions you may have.